Hello, and welcome to this video about my favorite molecule, DNA. Everyone has heard of DNA, which stands for deoxyribonucleic acid, but not everyone knows what it is and why it's so important for life. After watching this video, you will be among the elect few who actually knows what DNA is all about. DNA is a long molecule, a polymer, whose function it is to store information, and that information is crucial for the life of all cells in all organisms. In his book, The Language of God, Francis Collins, the former director of the Human Genome Project, the director of the National Institutes of Health, compared the information storage system of DNA to a language. Human language consists of verbal or written symbols that have meaning. Each word in a language conveys an arbitrary meaning, not connected to the sound of the word. In written language, words are composed of letters, which when combined in the right order make the words of the language. Again, the written words have no direct connection to their meaning, which is why we must learn languages. Written languages have rules related to the order of the letters. Most combinations of letters have no meaning. Here we can see one word among a group of meaningless letters. The spoken or written word tree, for example, in English, means a tall wooden plant covered with leaves. But there is nothing about the word tree that looks like or suggests a real tree. The only way to know what a tree means is to learn that the word is a symbol for the actual thing. The same principle is true for the information carried in DNA. It is therefore also a symbolic language, just like all human languages. The information in DNA is also coded in words consisting of letters, but for DNA there are only 4 letters, each of which stand for a particular molecule. You will see the chemical structures of these molecules later, but that isn't important. The molecules that comprise the DNA language are called nucleotides, or more commonly, bases, and their names are adenosine, cytosine, guanosine, and thymidine, but we generally simply use their initials, A, C, G, and T. All the words in the DNA language have exactly 3 letters, and these words are called codons. There are no spaces between the words in the DNA sequence, so every 3 bases form a codon. This means that where we start reading is very important. Here is a section of DNA with a sequence of bases, some of which are highlighted. If we put a space between every 3rd base, we can see the various codons. The first codon in this highlighted sequence is GCA, and the second one is TCC. So what do we mean when we talk about meaning in DNA? What do these codons, the words of the DNA language, mean? Each one means a particular amino acid should be put next in the process of making a protein. The entire subject of proteins and how they are made will be covered in another video, so I won't go any further here. Each codon signifies a particular amino acid. The meaning of the codons cannot be deduced from their chemical nature, which is why the language of DNA is symbolic. Here's an example. The codon ACT means, or codes for, the amino acid threonine. We can also see here the chemical structures of both that codon, ACT, and the amino acid it codes for, threonine. The chemistry of nucleotides and DNA is unrelated and entirely different from the chemistry of amino acids and proteins. This is why there cannot be any inherent chemical reason that the ACT codon should stand for threonine and not for some other amino acid. If we have 4 letters that can be arranged in groups of 3, then the total number of possible codons is 64. We know that every codon codes for something, but there are only 20 amino acids, so how does this work? To see that, we should look at a table of the genetic code, where we can see the definition of each of the possible codons. The table shows each codon in capital letters, and the 3-letter abbreviation for the amino acid that the codon codes for. What we can see is that several codons mean the same amino acid. For example, all 4 codons that start with AC code for threonine, and CAT and CAC both code for histidine. There are also 3 codons that tell the protein synthesis process to stop, and the codon for methionine is the start codon. I will be making at least one video about the incredibly complex and beautiful protein synthesis system, so I won't say any more about that now. Instead, let's take a closer look at the DNA structure and how DNA is able to replicate itself. This means it's time to let you in on a secret, namely the simple yet fundamental fact of all molecular biology. All of biology, from DNA replication to protein synthesis to gene editing and everything else, is based on this one simple fact. DNA is a double-stranded molecule, with the bases on each strand interacting with the bases on the other strand. And the rule is that an A is always opposite a T and only a T, while C is always and only opposite a G. The AT and CG combinations are called base pairs. What this means is if you know the sequence of one strand, you automatically also know the sequence of the other strand. Wherever you see an A on one strand, you'll see a T on the other, and wherever you see a G on one, you'll see a C on the other, and so on. Now, of course, in biology, there is nothing that is always true, and that goes for this fundamental rule as well, but we will discuss this more in the next video. Here's an example of a DNA sequence from one strand with the A's and the T's in red and the C's and the G's in green. We can predict the sequence of the other strand, since for every A there will be a T, and for every C a G. For every T there will be an A, and for every G there will be a C. To see how this works chemically, we can show 2 strands of DNA, one with an A and C, and the opposite strand with the complements T and G. As they come close together, we see the formation of the 2 base pairs, AT and CG. You will notice that the bases fit well, and that bonds can form between the atoms of each base with its complement on the other strand. Watson and Crick discovered by building models that the only way DNA could form a stable double helix structure was for all the A's to bond to T's, and only T's, and all the C's to bond to G's. This photograph shows James Watson and Francis Crick with their model of DNA that demonstrated its structure, and the base pairing rule that revolutionized biology. In their 1953 paper, they drew a picture of the DNA with the base pairing double helix at the top and a hypothetical, at least at the time it was hypothetical, concept of how the fundamental base pairing rule could result in the replication of DNA, and that hypothesis proved to be correct. What we now know is that during DNA replication, the 2 strands separate, and each strand serves as a template for the construction of a new strand, using the base pairing rule to produce 2 new double strands. Let's see how this works. We start with a double-stranded DNA molecule which separates to form 2 single strands. Then, new bases are added opposite each of the original strand according to the base pairing rule. So in other words, each A will attract a T, every T bonds with an A, each G is paired with a C, and each C pairs with a G. As these nucleotides find their complementary bases on the original strands, and they form base pairs, they also form a ribophosphate chain or backbone, just like those of the original two strands. The final result is that the double-stranded DNA molecule before replication, which is shown in blue, has become two identical double-stranded molecules, each with the same sequence as the original molecule, with the new strands here shown in black. And that is the basic story of DNA replication. It's also where we will end this video. If anything in this video was not clear, you can look up the details online, or, if you like, send me a question on Twitter, where I am @SciGart. And if you follow me, you can send me a DM with your questions, and I will try to answer them. So far, we have only scratched the surface. As we go deeper into the marvelous story of molecular biology and biochemistry, you will begin to see the divine design that is central to all of life. I intend to make several videos related to all the subjects that I cover in my recently published book, The Works of His Hands, A Scientist's Journey from Atheism to Faith. Please subscribe, hit the like and notification buttons, and stay tuned. Thanks for watching, and God bless you all.